when you take cell wall, assume that this is a plant cell. So a big vacuole is the indication of a plant cell. Okay. Then if you take cell wall around plant cell, the cell wall is again made up of various layers. Like the first one is the middle lamellum. So middle lamella is the outermost layer of a plant cell. Remember that one. Okay, outermost. So I'm insisting on the point outermost. Then you'll have a primary cell wall. Then you'll have a secondary cell wall. In some plants, you may have the tertiary cell wall. This one is an optional. Okay, all plants won't show the tertiary cell wall. So why some plants only will show tertiary cell wall, that I'll discuss. Now, when you take the plant cell, assume that this is the plant cell. You are having a middle lamellum at the outer side. This is middle lamellum. Then you'll have primary cell wall. So the orange one, I'm just showing a color difference. That's the primary cell wall. I'm showing a degree, 1 degree, primary cell wall. Then you'll have a cell wall inside to primary cell wall. That is the secondary cell wall. So I'm showing it in a purple here. Okay. Now when you take the secondary cell wall, the secondary cell wall growth occurs by accretion. Accretion. Then when you take the primary cell wall, the primary cell wall growth occurs by intersusception. Intersusception. This is it. So here when you take the whole cell wall structure, so the A, I'm just explaining first. Okay, so this is the plasma membrane. Okay, so I'm circling it with the orange pointer. Okay, so that's the plasma membrane. So here when you take a plant cell, this is the plasma membrane. So innermost one is the plasma membrane. And after that you are having secondary cell wall. I'm showing it in B. See, this is the secondary cell wall. So that's the secondary cell wall. Around the plasma membrane, you are having secondary cell wall. Then you are having primary cell wall above the secondary cell wall. This one. Okay. So this is the primary cell wall. And around which you will have the middle lamellum. So if you observe both A and B peaks, okay, the blue one or the purplish blue one is the outermost one. That is the middle lamellum in both the peaks. So this would be the middle lamella. Okay, so this is middle lamella, and this primary cell wall, secondary cell wall. Then inner to all the wall materials, you are having plasma membrane. Clear? Now here you are having various terminology for the growth of cell wall material. Intersusception growth you will observe in primary cell wall. Accretion growth you will observe in secondary cell wall. Okay. Now when you take the contents of middle lamellum or the primary cell wall or the secondary cell wall, few variations are there. Middle lamellum purely made up of pectins. Okay. Now you may have calcium or magnesium pectins. I'll show an example where the middle lamella application can be observed. After explaining this part, I'll get into that point. Okay. Now when you take the primary cell wall majorly will have cellulose and hemicellulose. Major portion of primary cell wall. Then when you come to secondary cell wall, you will have special type of materials called lignin, suberin. These terms I explained in the last class. isn't it? The primary cell wall you will find in meristematic tissues and some parenchyma tissues, where a secondary cell wall you will find in bark, or in some cases even in cork also you will find. That's why the bark and cork are very tough due to presence of those materials. These are polymers. I mentioned that word in the last class itself. Organic polymers. So the lignin and the pectin are organic polymers. This is all about plant cell wall. You won't find this kind of complicated structure in animal cell wall. Animal cells don't have any cell wall. Is it clear? Why plants only will carry this much complicated cell wall nature? Can anyone explain that? So here, as a father said, the environmental conditions, whether it's abiotic or biotic factors like the temperature fluctuations or the humidity or the sunlight variations, all these are the factors from which the plant has to protect itself, because plants are the immobile living organisms, isn't it? Like animals, they cannot go for shelter when the climate or the environment changes happens, like very high temperature or very cold temperature, or a high humidity rate or heavy rainfall, something like that. All these factors can be avoided by an animal being a mobile object, isn't it? But when you take plant, they are immobile and stationary at one place. So they have to get protection from all environment factors. So in order to protect themselves, the plant cells, all the cells in plant body are specialized in having a particular structure called cell wall. And again, cell wall, you are having different layers like the middle lamellum, primary cell wall, secondary cell wall, and a tertiary cell wall. Okay, so the to protect themselves from environmental factors, being immobile living organisms, plants only do carry cell wall other than plasma membrane. Don't forget that point. Plasma membrane is still there. Okay, plasma membrane is the innermost layer of a plant cell, and middle lamellum is the outermost layer of a plant cell. That's the difference. Okay. Now, when you come to this point of functions of cell wall, when you take the function of cell wall, the first one is nothing but providing shape and giving protection to the plant. So that's the primary function of cell wall in plant. And other than that, it gives mechanical support to the plant. See, during the strong winds also, so many plants will have the flexibility and the rigidity nature. They won't fall apart from the soil. They will be having strong root system under the soil. Even the stem also provides mechanical support. All this is because of the cell wall around each and every plant cell. Okay, giving mechanical support is the next function of a cell wall. And when you take this point, involvement in allowing some molecules to get into the cell and inhibiting some molecules to get into the cell. The movement of molecules in and out of the cell wall. When you come to this point, here we discuss about torosity. It's a new term. Torosity. Torosity is something related to concentration of solutions. What are the concentration of solutions around which a cell can survive, and a cell may get shrinked or swelling may happen. So all that is related to the cell wall, the most important function. Here when you take a concentration of solution topic, you're having some solutions by means of isotonic, hypotonic, and hypertonic. So in this diagram, we are having a term called counteract-Turger pressure. This point also we will discuss here only. So both these points are interrelated. Movement regulation by the cell wall and counteracting turgor pressure by the cell wall of plant cell. What is that? That we'll see now. Assume that this is the plant cell. Now, the plant cell surrounding medium is having a solution whose concentration is equal with the concentration of cytoplasm inside the cell. I'm using the word equal. Suppose if the outer surrounding solution or outer medium of a plant cell is having equal concentration as that of cytoplasm inside the cell, then that is called isotonic condition. Storosity word is nothing but the concentration of the solution in which the cell is present. Okay, so here the torosity is isotonic. Iso stands for same. So inside the cell cytoplasm, concentration in terms of pH if you take is equal to the outside concentration. In such case, the cell will be healthy. It won't show any drawbacks. Means the cell wall, no need to go for any protection for the cell because the outer concentration is same as the inner concentration. But the problem comes when the cell is there around which the concentration is assumed that low. So the outer concentration of a cell is low compared to inner concentration. So what happens? Always there is a tendency of flow of materials from high concentration to low concentration, isn't it? So what happens? Whatever the cytoplasm or whatever the cell contents inside the cell will move outwards. This results in shrinkage of cell. This we discussed in guard cell activity in stomatal complex and tissues chapter if you remember. Okay, so here what happens as a result of outflow of the materials from the cell to the surrounding medium, because the surrounding medium is having less concentration. Such condition we call as hypo. Hypo stands for what? Low, isn't it? So if the outside surrounding environment of a plant cell is having low concentration, hypotonic solution, then the cell shrinkage happens. The cell gets shrinked because all the contents of cytoplasm and other cell organelles will try to move out from high concentration towards the low concentration. The exact opposite happens when it is hyper. Hypo means what? More amount of concentration in a cell. So here comes the explanation of turgor pressure. Assume that this is the cell around which you are having a cell membrane. Okay, so here when the surrounding solution is having high concentration of molecules, using the high concentration of molecules around the plant cell. In such case, what happens? The tendency of movement of molecules from high concentration to low concentration happens. As a result, inflow of materials from outside to inside happens. Inflow. Quite opposite to hypotonic solution. So this is called hypotonic solution, outside environment. So because of the inflow of material, resultant of this condition would be swelling of the cell, isn't it? Something gets inside of the cell, it tends to get swell up, and it may result in bursting of the cell and ultimately death of the cell. But this condition will not happen in case of plant due to presence of cell wall. What happens here? Because of the outside hypotonic high concentrated solution, inflow of materials happened and the cell got swallowed up. The swollen cell might be looking like this. Okay, because of the inflow of materials. But around which you are having a plasma membrane, isn't it? So this is the plasma membrane. If you observe this peak and this peak, here there is some space between the plasma membrane and cell wall, isn't it? But due to swelling of the cell, the space reduced. This is called turgor pressure. triggering the increased pressure. Trigger nothing but triggering the increased pressure. Because of this increased pressure, the cell wall will put some pressure on the plasma membrane, and plasma membrane itself will show some other pressure opposite to this trigger pressure. So this is how the bursting of plant cell wall will not happen due to presence of cell wall, even though it is present in hypertonic solution. But when you compare with animal cell, the cell will burst out due to absence of cell wall. So these are the different functions of cell wall.